Hi Hannah.
Hi.
How are you going?
Very well, thanks.
So we've just been with Jane, and Jane has talked about collecting samples in the wild, and she's got 2 packages of samples that she's brought back to you. What happens next?
Right. So the field necropsy investigation has been conducted, and that really is only the first part of the job. We've gone out, we've looked at the animal, and sometimes we know what's wrong or what has caused the disease, caused death, based purely on the gross examination, but often we have more questions, and that's when we bring the tissues back to the lab to complete our examination. So we'll have frozen tissues that we put into the freezer, and those can be used for microbiology, for toxicology, for asking all sorts of questions down the line. But what we do with the formalin-fixed tissues is prepare them in small cassettes so that we can have them processed, and I can eventually look at all of these tissues under the microscope at high magnification and start to interrogate at a finer level of detail what has actually gone wrong with that animal.
We used the whale as an example. Can you tell us about some of the other samples that you've got there?
Yeah, so we've got a few mortality investigations going on at the moment. So you can see that we've started with an animal that is as large as a whale, or perhaps a long-nosed bandicoot or a ring-tailed possum. But by the time they come to the lab, they're actually trimmed down into small sizes so that we can actually get them processed and looked at under the microscope. So yeah, we've got quite a few things going on. We've got a few ring-tailed possums here.
And what would they have come in with?
Those ones were actually in care, so they were juveniles that didn't do well and were unfortunately found dead by the carer. And so rightfully they're very concerned about what might be going on and whether there was any infectious diseases they need to be worried about in the other animals they're caring for, or if there's something going on in the environment.
So up the back there, I can see, does it say currawong on there?
Yeah,
we've bird?
We've got a currawong, we've got a green sea turtle, we've got a boobook, we've got all sorts of things. So we see a real diversity of species coming into the registry.
And when you prepare it, what do you have to do to get it onto a slide, the finished product?
Yeah, so I can actually show you. What we actually do is pour out the tissues, and then I'm literally going through organ by organ and cutting them into small enough tissues to fit onto this cassette here.
Yeah.
So, happy to oblige.
Go on, give us a look. This is proper fantastic investigation work that goes on, and it's critical to the health of our wildlife populations in Australia and around the world for that matter, Hannah.
Absolutely is. And, you know, when we go into the field or we conduct these necropsy investigations, our immediate concern is the cause of death in that individual animal, but all of these samples form part of our archive. So we can actually interrogate tissues going back 20 years and start to ask questions about what has changed over time, whether it's something that has changed in the environment. If we're seeing a new and emerging species, we can go back and look at tissues that came into the registry 15 or 20 years ago and start to identify temporal patterns. And that's really critical with species we know very little about, particularly the marine mammals.
Yeah, right. So you've just drained that.
Yes, I have decanted our tissues and the formalin is now removed. So this is one of a group of ring-tailed possums I examined last week. And now it's really just a matter of me going through and isolating all the bits and pieces that I need to look at to complete my investigation of this animal's death. So we've got, they're all very little. We've got kidneys there, that's a piece of lung, another piece of lung here, some liver, more liver, that's the intestinal tract, that's the heart, and then the brain, which I usually do
incredible.
last.
Quite incredible. And this work is critical to the survival. I find it fascinating. I think one of the things about Taronga TV is that we get to come into these beautiful places to talk to these amazing scientists, and thank you very much for your time.
Oh, my pleasure.
I can feel many more stories coming on, folks, from here, that's for sure. And I think, you know, as I said, it might not be everyone's cup of tea, but this is incredibly important work, critical work that is going on for the survival of Australian species and species around the world. And the scientists talk to each other. It ends up coming into our labs here, and they have 20 years worth of tissue samples in our zoo here. It's quite incredible work. Thank you, Hannah. We'll be back with Taronga TV and more stories from the Australian Registry of Wildlife Health. It's critical work that's going on. You never know what's around the next corner. We'll see you soon. Thanks, Hannah.